again, I want to thank you for inviting me to give this lecture. I always enjoy talking to students who haven't made up their minds about what they want to do. And it's always a challenge and a privilege to be able to address an audience that may or may not know anything about biology, and to be one of the first people to try and convince them that there's something of interest to it. Could it be possible to lower this kind of slide? Okay, that's great. Okay, so over the coming hour, I'm going to start this set of 3 lectures. And the way I plan to do it is, rather than lecture on a single topic, what I'm going to do is give you a spread of different topics that I think make biology interesting. They happen to be topics that I've actually done research on, but also I think it does demonstrate the spread of what's in biology. Now, I know you're all physics students and are all looking for me to solve some complicated equation on the board, and I'm not going to give you the satisfaction. What I'm going to try and do is to give you a flavor of the open questions in biology. Somebody says open questions, it means what are the big mysteries and what are the things that as scientists we would like to solve. But I will try and hit that math button a little bit, because I'll show you in each of these lectures, you'll see buried in there, I'm not going to make a big deal of it, but buried in there you'll see that the way we solve these problems is actually by making use of very sophisticated tools from mathematics. So in the same way that mathematics has been transformational in physics in the previous centuries, mathematics is currently transformational in biology. And this is one of the reasons why physicists entering biology often enjoy great success, solving problems that people who come before them are not able to solve. So it's mathematical intuition or insight. Okay, so let me, without further ado, start. Here are the things I'm interested in: this little triangle, cells, information, and evolution. And the reason I'm interested in this is because if you sit and think about it, if there is life on other planets, then almost certainly it's based on information and evolution, and there are very strong arguments to be made that it's based on cells. The point I'm making is these are universal aspects, we think, of what life should be like, and these are the kinds of things, these are the kinds of big questions which I like to sit and think about. And then of course you have to pick a smaller question for actually solve. Just for a bit of background, as you already heard from my introduction. So my PhD is in physics, and the last time I studied biology was in the 10th standard. And in fact, back in the day, the textbooks were so bad that the last bit of biology I studied was simple leaves, compound leaves, this kind of stuff. Right? So I never really learned any biology. No. And I picked it up late in life. And I think that's actually a very good way to do it. Right? If you're put off by biology now, don't worry about it. Come back to biting later. These are all the different things I managed to work on. And just the reason I put this up over here is because, you know, I'm not particularly exceptional in the world of biology, having diverse topics like this. In biology, you can actually be sitting around, you can be sitting in a bus, you can be reading a book, you can just be watching a movie, whatever it is, and suddenly realize that there's some question that somebody is not solved. Okay, so that doesn't happen very often in physics. So because physics is old and all the big questions have been solved. So you can come up with questions. And then the next day, if you put your mind to it, you might actually solve the question. And that's how many open questions there are in biology. And so the fact that I worked on things like randomness, bacteria, transcriptional networks, which I will talk about in the 3rd lecture, synthetic biology, evolution eukaryotes, all these things. It's really what makes biology exciting. It's just that you can just look around and you can find something new that nobody has seen before. The Simon Center was mentioned during the introduction. I just mentioned what that is. Oh, by the way, biology is also fun because it gives me the chance to work with other kinds of people who are not scientists. So I have a big art-science collaboration. So I mentioned briefly in the 3rd talk. If any of you are interested in these kinds of things, I'm actually a big believer in spreading science beyond the science world. So if you're interested in this kind of stuff, please come and talk to me about it. There's a lot of different ways that we do this. Okay, the Simon Center is just a little place inside NCBS. It's a place where a lot of people, many of whom are physicists, get together to do research in all kinds of aspects of biology, ranging from single molecules to cells that I work on, big organisms like humans and flies, and entire ecosystems like the savanna. So really the spread of scales in biology is huge. And because there's so many different things that we work on at so many different scales, we need physicists to come and do biology. So not just physicists, computer scientists, engineers, mathematicians, and so on. One of the ways we do this is through this monsoon school. I don't know if you've heard about this, but every summer we run a 1 or 2 week school where we bring in students from physics and we teach them all kinds of interesting problems and methods that they can use to address biology questions. So you're all welcome to look up the website and see come to that. It happens every summer. Okay, so let me start. So I was asked to give a series of 3 lectures, and I was asked to give a title for this, so I thought, life is computation, and past, present, and future. Past is today's lecture, present is tomorrow, and future is the third lecture. When I say life is computation, I'm not going to give you a lecture on what a Turing machine is. How many of you have heard of Turing machines? Yeah, lots of you, right? Now, I'm not here to give you a lecture on Turing machines, although it's in the title, the abstract on the poster. I'm not here to give you a lecture on how computers work. I'm here to give you 3 example problems, and it's up to you guys to figure out in these examples where the computation angle is. Right? So in these little question answer sessions at the end, I'd like you guys to extract where in this topic there's some aspect of computation, and we'll discuss it at the end. So it won't be a formal part of it. Okay, so today's topic is the past, and it's really a very distant past. It's 1 or 2 billion years past. And I'm going to tell you about something called molecular archaeology, and how we use that to hunt for the earliest types of cells. If there's some word on here which doesn't make sense, like a biology jargon, sometimes I forget that people don't know it, please stop me and ask me. So here's the word story, in the title of the talk. Actually, it's called search, poster announcement called story. So whenever people give a talk in biology, many times they say, I'm going to tell you a story. And this puts off some people, right? Because they hear, they want to just hear a story. They want to hear something a bit more bigger. So you have to ask, what is the difference between a story and science? Right? Is science a story? Very simple way to think about it. So a story is something that happened only once. Right? I'm going to tell you a story. Here's how it happened. Science, a very good definition of science, is something that happens many times. Right? We typically call them laws of nature. If something can happen reproducibly, it can be experimentally tested, and then you can come up with a theory for it. If something happens only once, what does that mean, right? Is it really science? And so, for generations, people have been convinced, at least in the past century, that anything that happens only once, that's just a story, right? Things that happen lots of times, that's science. And I want you guys to step away from this aspect of thinking. Here's a bunch of questions. Where do we come from? What are we? Where are we going? You would all agree these are big questions, right? They keep people up at night, mainly philosophers. Not the rest of us, but does that mean they're not worth asking just because all this happens to do with story? Where did we come from is a story. It happened only once, it happened in one way. And often people will say, denigrating, that is not science. Right? That's history, or some such thing. And I want to point out that it indeed is a scientific question, if you can ask it in a scientific way. And so the question of where we came from should occupy all your thoughts. I mean, it's one of the great things that we now know, in many ways, how the early universe was made, how the earth was formed. We don't know how life arose, but once life arose, we know how it evolved, we know about recent human evolution, we know about the evolution of the brain, we know all these things. And that's amazing. This is the first time in human history that we've known where we come from. At least we filled in a lot of the gaps. So these are scientific questions, they should occupy our thoughts. Incidentally, these 3 questions actually come from the title of this painting. I don't know how many of you have seen this painting. It thinks of it, but it's a painting by Gauguin. It's called Where Do We Come From, What Are We, Where Are We Going? It's a very beautiful painting, but I'm only using it because it brings up these sort of questions. And as it happens, the 3 lectures I'm going to cover where do we come from, where are we going, and sort of when I say what are we, you'll see what that means. So now I'm going to tell you one of these crazy facts in biology that we're going to try and explain. Here's the fact. Eukaryotes and prokaryotes. Now how many of you have never heard of these words? Okay, very good. There's at least one person who's never heard of these words. Many of you have already heard these words, right? You must have heard it in some textbook or some terrible thing like that. Yeah. When do you think these words were put in the textbooks? Which decade? Was it the 1850s? Anybody say before the 1850s? Even though they weren't proper textbooks. Okay, everybody thinks it's after the 1850s? Anybody thinks it's after the 1850s? Raise your hands. Okay, it's either one or the other. So everybody else thinks it was exactly 1850s. Okay, how many people think 1900? How many think after 1900? Okay, how many people think after 1950? How many people think after 1960? Okay, it was in the 1960s. This is very new stuff. Okay, it was after the discovery of DNA and all that good stuff that you've heard. These words weren't even in the textbooks. Okay, why did it take so long for people to discover prokaryotes and eukaryotes? It's because it's an experimental fact. It's not something people just make up. Which experiment did they have to do? They needed an electron microscope to look inside cells and realize that there's one type of cell and there's another type of cell and they don't really look the same. The electron microscopes weren't really around, obviously, before 1940s or 50s, and they weren't really applied to biological samples till the mid-50s. And then, of course, it takes 10 years for something to reach the textbooks. Okay. So before electron microscope, people thought there were two types of cells. There's plant and animal cells, and there's other types of cells. In terms of that is not the correct classification. There are prokaryotes and there are eukaryotes. And it's amazing that this kind of crazy fact remained unknown for so long. Very new. Just to give you some context for the time, these are microfossils. There's no scale bar here, but that thing will be a few microns long. Yeah? And 3.5 GA means 3.5 giga years ago. 3.5 billion years ago. So there are fossils of living, by various chemical and morphological characteristics. You can figure out that these are probably of living things, probably bacteria. And they've been around for 3.5 billion years. How old is the Earth? The Earth is about 4.5 to 5 billion. The solar system is only 5 billion years. The universe is only 14.5 billion years. Yeah. So 3.5 billion years ago, a significant fraction of the lifetime of the universe, there has been life on Earth. But these are bacteria. If you fast forward a couple of billion years to 1.5 billion years ago, the fossils in the rocks change their nature. You go from these very small, morphologically not nondescript fossils to these very, very complicated, beautiful structures. Okay? This scale bar is now something like 10 microns. These are 20, 30 microns. Big cells, lots of complicated internal stuff. By the way, how do you think they find these fossils? There are graduate students who go around digging in rocks. Okay? Then they'll take the rock and then they'll send it, slice it, electron microscope, sorry, no fossil. And you go back and you keep on doing it. Right? These are called microfossils. And you can imagine how hard it is. This is real archaeology. It's not molecular archaeology, it's real archaeology, but very small. These are the reasons we know that prokaryotes were earlier than eukaryotes. These are the reasons we know that eukaryotes probably came around about 1.5 to 2 billion years ago. And these are the reasons we know that the old eukaryotes, these guys, are not quite the same as the new eukaryotes, us. Yeah? It's pure fossil evidence, the best kind. Unfortunately, as you can tell, cells do not leave too many good fossils. Very difficult to do. So we have to somehow reconstruct history without relying on too many fossils. And how do we do that? That is what molecular archaeology is. And that's what I'm going to talk about. What do we know? How did these 3.5 billion year old prokaryotes become these 1.5 billion year old eukaryotes? Eukaryotes must have come from prokaryotic ancestors. They weren't invented from scratch. How do we know that? Because they use a lot of the same machines. So, what do we know? Okay, anybody know who this is? A person named Carl Bose. If you don't know who it is, you should really go and read about it. Amazing man. Carl Bose, in the 1970s, decided that he was tired of his colleagues telling him about classification things, telling him that this is some type of insect and that is some type of diatom. So he didn't want biology and the spread and diversity of biology to be left in the hand of the professional taxonomists. You know how they say war is too important to be left to the like that. So he wanted some systematic way to go out there and say this is the diversity of life, without having to do all this measuring the length of the antenna and the number of spots in the foot and stuff. So he decided, let's just use molecules. This was post the DNA era. So he went out and he collected samples from all kinds of different places. And then he did something called sequencing. If you've not heard of sequencing, I can quickly tell you, if there's any DNA or RNA sample, you can send it through a sequencing machine and it will come out with a gel like this. You can read that gel and replace that gel with some letters, A, T, G, C, in some order. And that contains information for you. I'll talk about that in the third lecture. Nevertheless, every different species on the planet has a different signature. And in this way, Carl Bose did the first, what was called metagenomic experiment. He went and got sequences from thousands of species. And he said, who's closer to whom? That is what I'm going to call a species. If these two are slightly related, I'm going to call them related species. If they're really distantly related, I'm going to call them distant species.
So,
yeah.
Did you say this was pre-DNA and...
No, this is the post-DNA. Yeah? Because it was the 1970s. DNA was already up and about by the 1950s. Okay. DNA was up and about 3.5 billion years ago, but our knowledge of DNA was up in the 1950s. Okay. So what he found, and sorry, this is not very visible, but when he made all these sequences and he tried to classify them, he found that they broke into 3 different groups. Okay. What was he expecting? He was expecting 2 different groups, prokaryotes and eukaryotes. And indeed, he managed to get eukaryotes. But the prokaryotic branch split into 2 pieces. So you know what happened? This is just the 1970s. He had found an entire new domain of life which nobody had suspected existed. They were called the archaea. In fact, he named them the archaea. This is just the 1970s. Can you imagine? That in the 1970s, we discover an entire new domain of life that nobody, not a new species, an entire new domain of life that nobody suspected existed. Just like the electron microscope gave us prokaryotes and eukaryotes, the sequencing revolution gave us bacteria, archaea, and eukaryotes. So this makes the story even more confusing. Right? Now there's 3 things to explain. What is going on? So I'm going to cut a long story short. If you do a lot of the type of sequencing analysis that Carl Voss did, same type, but much bigger data, much less error, much more sophisticated samples, then you find the following very interesting. And here I'm just going to summarize a huge body. Here are the eukaryotes, they're in green. Here are the archaea, they're in blue. Here are the bacteria, they're in red. What you can do is get genes for each one of these things and get sequences. And then just plot them on some sort of grid. Each one of these little graphs is for a different type of gene. Okay? And 2 things are close if the sequences that they come from are close. And also what you will notice here, if you stare at it a little bit, is if you have green, blue, and red, most of these things fall into one of 2 patterns. Either the green groups with the blue, like here, or the green groups with the red, like there. Yeah? What that means is that we, the eukaryotes, we are nothing but bacteria plus archaea. Our genes either come from bacteria or they come from archaea. They don't come from anywhere else. Okay? So this should really worry you. What does it mean to say that we are a mix of different kinds of genes? Right? Nobody in biology says that kind of thing is allowed. I can't take, a cow and a butterfly and put them together and get sort of a fly. Okay? Or even a cow with wings. You can't do that. So what does this mean? So I'm going to tell you. What are those structures? These are just graphical ways of representing a huge amount of data, but every point is a particular gene from a particular species. And the little graph corresponds to a bunch of points from a bunch of species, and they're close together if the gene sequences are very close. It's just a cool graphical way of representing a huge amount of data. Okay? These are not real structures, they're just abstracts. It's another example of mathematics making things visible in biology. Yes? The gray are just the lines that indicate that 2 things are connected to each other. Yeah? Strong connection. Okay, so we are bacteria plus archaea. How does this make any sense? Here's another picture, same color scheme. Okay? These are human cell lines. This is just literally on a microscope. And you stain it, that means you add a certain fluorescent dye. And there are 3 dyes I put. This is just general experiments that people do every day in the lab. One dye just stains the overall cell. That's the green eukaryotic cell. One dye stains the DNA in the nucleus. You know the eukaryotes have nuclei, right? That's the blue stuff. Another dye stains every other type of DNA in the cell. You see it's over there, right? Why is there any DNA in the cell which is not in the nucleus? Right, there is stuff in our bodies which is non-human DNA, in a sense. Right? They live outside our nucleus, but inside every one of us. This is also quite mysterious. Now it turns out that those little guys are the mitochondria. Where did mitochondria come from? They came from the outside. They used to be bacteria. And these bacteria ended up penetrating and living inside another cell and taking up a permanent residence inside that other cell. That's what made the eukaryotes. That's why eukaryotes were. So what type of cell was the other cell? Must have been archaea, right? Yeah. This fact I'm telling you is very new. We've only started to realize this in the last 2 or 3 years. So mitochondria, which were bacteria, went into some other cell, which was an archaea, and became eukaryotes. And where did this happen? It's looking very much like it happened in some deep sea vent off the coast of Iceland. but I'll tell you more about that later. Maybe more than that. Okay. So now we're very happy and convinced with the fact that we are a combination of mitochondria and archaea to make eukaryotes. You know how we know this? This is a story, right? I just made up a story for you and you all believe it. You should say, where's the evidence? Yeah? I'm going to present one example of what we did to find evidence of this kind of thing. But who discovered this thing in the first place? This lady. Her name is Lynn Margulis. And in the 1970s, she was a contemporary of Carl Woese. She fought with the scientific community to say that mitochondria were actually free living bacteria which live inside eukaryotic cells, no matter how crazy that sounds. And here's the evidence, and here's the evidence, and here's the evidence. And eventually people believe it. Anybody know who this is? Anybody know? It's Carl Sagan. Yeah, Carl Sagan and Lynn Margulis were married. And Carl Sagan was very much interested in the origins of life. And so was Lynn Margulis. It's no coincidence that she came up with this very crazy idea, which eventually turned out to be true. Imagine the conversation they must have had. This is the photograph. This is a photograph taken on their wedding day. Yeah? Carl Sagan was a professor of astronomy in Bonn. One of the best popularizers of science ever. Yeah.
Yes, mitochondria give energy to the cell, right?
They do, but cells have their own sources of energy, right? So many archaea can have their own sources of energy without eating mitochondria. Good question. We rely on mitochondria now for that. So the reason mitochondria came in was not for energy, but that's a subject for another.
So the powerhouse of the cell is not correct. Mitochondria is the powerhouse.
They are now, but that's not why they first came. I'll tell you why they first came. Listen. Okay, so now.
So we have started relying on mitochondria and not on the source for the archaea. Why did that?
We don't know. It happened. It's a good question. Somebody should work this out. Yeah, so you see, he just came up with a question. Nice. Okay. I'm actually going to give a research talk, and unfortunately, when you give a research talk, there's some jargon. The jargon is absolutely terrible, right? This is part of the reason why people don't like biology. No, what do you about it? Once I tell you what it is, you know what. Here's an example of a tree. Does everybody know what a tree is in the context of biology? Not this tree. What is a tree in the context of biology? A tree is just a thing that keeps on branching, right? Yeah? And here I've drawn a tree which is actually a long time. On the x-axis I have billions of years ago. 1,000 million years ago. 2,000 million years ago. 2 billion years ago. You know what's so cool? We used to keep saying billions and billions. You know what was that? Carl Sagan. He had this TV show called Cosmos and he used to keep saying billions and billions. The reason he got away with that was he was an astrophysicist. Astrophysicists always get to say billions and billions, talking over tea and saying billions and billions. But biologists can also say that. Right? And that's what's really cool. You can actually get in and do a piece of research that has something to do with things that happened billions and billions of years ago. While sitting in front of your computer. So here's the trick. This thing here is called the last eukaryotic common ancestor. It is the ancestor of us all. Okay? But it's only our most recent ancestor. It's not the first eukaryote. It is the last eukaryote that is still the ancestor to everybody. Who is everybody? Here's some terrible names. Some you'll recognize. Fungi, everybody recognizes. What's an example of fungi? Yeast is an example. Metazoans, you may not have heard that word. Metazoans means animals. I don't know why they don't call them animals. It's called metazoans. This is an example of a metazoan. I could have put humans, but I like this. It's a cuttlefish. Okay. Amoebozoans. Amoeba you might have heard of, right? Amoebozoans. Green algae you might have heard of. Right? One branch of green algae are the plants, okay, which are also. Then there's all kinds of other things. Alveolus, raminophytes, all kinds of stuff. Each of these you would have seen and heard of. These are called, like paramecium is an alveolus. A diatom is a raminophyte. Trypanosome, because of sleeping sequence, is an excavator. The point is that all these things you see in the textbook, don't think about them like that. Think about them like this. Fungi and metazoans are the closest cousins. Green algae and red algae are the closest cousins. Then the next closest cousins are like the diatoms. And these are distantly related. 1.5 billion years ago for everybody. This is how you should think of it. We live over here. So most of the things biologists study are just over here. But this is all the stuff on it. These are all the kinds of eukaryotic cells we know right now. Yes. Eukaryotic cells. I'm not even talking about the bacteria. Right? The bacteria and the archaea are two completely different things. Viruses are not alive, they're not cells. Viruses are just little bits of DNA. Okay. Yes. That's exactly what it is. remember the structure of this tree, remember that we are here, remember plants are here, remember the time scale, and two other things. Not all life is evolving on a tree. Sometimes two branches of life come together to make another branch. We already saw that mitochondria came into some cells to make eukaryotes. Mitochondria were bacteria. This happened again, surprisingly. Another type of bacteria, which is called cyanobacteria. Blue-green algae is also called. They do photosynthesis. And the blue-green algae ended up inside another eukaryotic cell, and they became the photosynthetic eukaryotes. It's amazing. Right? They became chloroplasts of the photosynthetic eukaryotes. Still, the most organisms that do photosynthesis are the blue-green algae, cyanobacteria. Plants contribute a fraction of what the algae do, of what the bacteria do. But nevertheless, here's one amazing thing that happened. Here's another amazing thing. These creatures are called endosymbionts. They live inside another creature in a symbiotic relationship. My whole talk is about endosymbionts. Mitochondria is an endosymbiont, chloroplast is an endosymbiont. So here's the timeline I'm talking about once again. That is today, somewhere there, present day. Right? Probably far off the board. Yeah? This is the past. This is 3.5 billion years ago. Remember, there's the prokaryotic ancestor in the fossils. By around 1.5 billion years ago, you already had eukaryotes. Okay? Something must have happened to go from prokaryote to eukaryote. Now we figure out why mitochondria came into it in the first place. 2.5 billion years ago, something interesting happened over. There were bacteria. Bacteria used to rely on chemistry, on deep sea vents to survive. They didn't rely on the sun, at least not directly. But 2.5 billion years ago, one group of bacteria, the cyanobacteria, invented photosynthesis. Right? And when they did that, what happened? They made oxygen. And they completely transformed. It was the first big wave of global climate change on the planet. They completely transformed the atmosphere. It was filled with this poisonous gas, oxygen. And one of the first things mitochondria did is they provided a way for a cell to quickly use up oxygen so that it wasn't poisoned. That's part of the reason why we have mitochondria. How they invented photosynthesis? Through this stunning complex process of heavy. That's the only answer I can give you. They tried many things. Somebody invented photosynthesis and they survived. I'll talk about evolution tomorrow. What are your questions? Okay. So, global oxygenation. After that is when mitochondria entered eukaryotes. After that is when the eukaryote became complex. Incidentally, eukaryotic genomes are much bigger than prokaryotic genomes, but I won't have time to discuss.
How is the transition between not having the ability to undergo photosynthesis
Yeah.
and having the ability to do? What's in between?
Well, obviously a series of intermediate steps, and we'll talk about that in a second.
So genomes.
Genomes?
Genomes.
Sorry?
What are genomes?
What is a genome? A genome is the entire set of DNA that's contained in your cell, in any one of your cells. And it contains information to make it. Yeah? Everybody fine? Good. So now that I've given you the background, I'm going to spend the remaining time in this talk to give you, you've all now convinced there's some really big questions out here. So now, confronted with a big question like this, how do we even address such a question? We have to make it a scientific question. So here's a moderately scientific question. Imagine you're a bacteria and you went into some other cell. And this is the ancestor of the mitochondria. So you have to have some way to decide that this primitive partnership becomes a permanent partnership. Right? There must be some series of events, right? It's not that immediately the mitochondria is happy living inside another cell. The cell will treat the mitochondria as some sort of infection. So it's not a very nice partnership to begin with. What's the first thing that must happen? Mitochondria divide, right? They're also cells. The host cell also divides. The mitochondria divide faster than the host cell, they will just burst. The mitochondria divides slower than the host cell, what happens? Eventually the host cells will lose the mitochondria. So you can't have a permanent partnership. So somehow the mitochondrial division has to be synchronized with the host cell division. Right? That won't happen right at the beginning. How did that happen? So that's what we ask. What did the early mitochondrial division apparatus look like? There is a scientific question. It's slightly more specific based on the big question I covered earlier. We're trying to fill in the intermediate steps in this crazy process. Here's some questions about reconstructing ancestors. This little thing you see here is actually a little cutout of a much bigger tree, which I'm going to show you later. It's a tree. It's a tree where time is going upwards. It's the tree of, let's say, animals. Two branches in this tree are the humans, which are the genus Homo, and the chimpanzees, which are the genus Pan. Okay? Now I'm asking you a simple question. what did the ancestor of these two guys look like? And I'm giving you three options. Is it this, or this, or this? How many people think the answer is the first one? Okay. How many people think the answer is the middle one? How many people think the answer is the last one? Nobody thinks the answer is the last one. Okay. Let me pause and tell you something. Look, this answer is almost certainly correct in the sense that we know through a lot of different lines of evidence that all animals that are vertebrates came from ancestral states that look a bit like today's numbers. Okay? So this indeed is an ancestral. But really that's not the question I was asking. Right? So I could have put bacteria here also, that would be a correct answer. Right? So you were also ancestral to us. We're going to have a big discussion to see what the ancestors are. And that never ever happened. Scientists do not talk to each other. Right? So how do we reconstruct ancestry? So the answer must be that look, let's decide there's some character, trait that we can all focus on and become experts on. Right? And then talk only about ancestry based on that trait. That way we're learning different kinds of biology. If we had to do that, which trait would you use? Is it hair or no hair? Clearly not. Would it be brain or no brain? No. It's so complicated. We don't even know what the brain is. Right? How can we figure out our ancestry? So what trait would you use? Cells. What about the cells? Cells is cells, right? We all have cells. DNA. Okay? So, the idea is the following. Somebody brought this up. It's hard to do statistics on these very hand-wavy traits. It's easy to do statistics on DNA. Why? Because DNA, RNA, proteins are totally digital. It's just ATTC, right? So we can use every method that's been applied to any kind of digital transformation to work out the ancestry of sequences. This is what I'm going to show you the story about. And so as long as we decide there's some sequence, that's good. We can reconstruct its ancestry. But then, we're going to use that sequence to say something more interesting about biology. Here's an example where people look at the sequences of the flu. Right? And you can see that the sequences change. This is time going to the right, and sequences keep on changing. This is the current epidemic, next epidemic, next epidemic. You would like to know all the flu variants that are around today, which is going to be the next one, so that I can make a vaccine. Turns out you can use sequence data to predict that before the event happens. In fact, WHO is starting to use this to make the vaccine for the next flu epidemic before it even happens. It works. So the trick is to find a molecular marker which can give you a hint about something you're interested in. And my favorite example of this is the following thing. That's a t-shirt. When did humans start wearing clothes? How do you work this out? Well, you could look for archaeology, you could find dig sites where they had clothes or no clothes or whatever. But of course, clothes are perishable. It's not like you're going to find evidence of clothes. It's very, very old. So, this group decided to look at, I think, what were they, lice or ticks? Oh, I forget. It was lice. Okay? We have two kinds of lice on our body. We have body lice and we have head lice. As it happens. Okay? You may not have known this, it's an awful fact. But we have head lice and body lice, they're two different kinds of species. Okay? This guy sequenced the head and body lice DNA. And they found out that at some point in time, they were actually the same. So at some point in time, we had just one type of lice on the body. And then it became two separate types of lice. The body lice, which lives on clothes, more or less, and the head lice, which lives on it. And they used that time point from the DNA to date the invention of clothing. Okay? And subsequently, it has been found that archaeological dig sites also agree with this particular. So isn't this beautiful? You need to find some sequence data which you can use to answer some completely different questions. And then using the power of mathematical statistics on the sequence data, you can answer the complicated questions. Yeah? Alright. So here's the question. What did the early mitochondrial division apparatus do? I've already told you it's a good question to ask. Now all we have to do is to use a protein or a sequence to answer that question. Have a question?
So the previous one, it was accurately what order?
So these errors are at that time scale on the order of 10%. But it's not bad at all. Yeah?
What's the figure for?
Oh, what's the figure? Some tens of thousands of years. It's there on that part. Okay. So which protein should be used to figure out something about mitochondrial division? Here is where none of you know. I don't know either, right? So what do? I walk down the hall to a colleague of mine and say, how do mitochondria divide? They'll tell me here's a bunch of proteins. Right? And then I pick one of them. Okay? So So here's how mitochondria divide. These are mitochondria. There's a big cell here, which I haven't drawn. Inside the cell, the mitochondria live. The same way there's a big cell inside the cell, chloroplast. Right? Mitochondria divide by being squeezed by a ring of polymeric protein, which is called dynamin. I'm just telling you this. It's a fact. Chloroplasts, so also endosomes, also divide using the squeeze of a different version of the same protein, dynamin. It's like dynamin A and dynamin B. How do bacteria divide? If you have to be squeezed from the outside, bacteria can't be squeezed from the outside. All their stuff is on the inside. So it turns out bacteria divide by being pinched from the inside by a completely different protein, which is called FTS. Okay? Bacteria divide using that. Mitochondria divide using this. Interestingly, chloroplasts happen to also have FTSC on the inside, which is a very strong indication of their bacterial origins. But mitochondria don't have. Okay? What do we do about it? Yes?
I was thinking for the reason for mitochondria to be inside the cell, would it be that cells used to eat mitochondria and somehow they couldn't digest it?
Could be. Could be. It's more likely that mitochondria infected the cell and then they just stuck around. Yeah, so we can tell the difference between these two cells. Okay, so I've just been talking about division, right? So in the next 5 or 10 minutes that I have, I'm going to finish the story. This is how things divide all across life. Okay, this is the last universal common ancestor. These are all bacteria. The bacteria divide from the inside using this protein called FTS. I'm only interested in the mitochondria of eukaryotes. The mitochondria of eukaryotes divide using this thing called DRP, dynamin related protein. That's all I wanted to know from this. So here's the problem, right? We're asking what the ancestral mitochondrial division apparatus was like. Just like the problem with human evolution with many traits, dynamins do many, many different things. And when we first confronted this problem, we thought we solved it very quickly. We took dynamin, we said we're going to reconstruct ancestry, we made a phylogenetic tree, we started looking at it, and boom, we were big trouble. This is another type of phylogenetic tree. I'm not going to spend much time on it, but here, time goes out radially. You start off there, which is the center of the tree, and time proceeds radially outwards along every one of these branches. For the moment, you can ignore these net-like objects on the inside. We found out that there were really many types of dynamics. There's one entire group that does chloroplast division. There's another entire group that does mitochondrial division. And a related group that does vesicle incision. I'll tell you what that is. If you have a eukaryotic cell, you need to sample things from the environment. They do that by pulling in a bubble, which is called a vesicle. That bubble has to be pinched. Just like mitochondria pinch, that bubble has to be pinched. That's also done by dynamin, but a different dynamin. Some dynamics are antivirus dynamics, all kinds of stuff. So what do we do? How do we reconstruct the ancestor? And I'm going to tell you, in the meantime, about something called living fossils. This is the idea of molecular archaeology that allow us to really rigorously reconstruct what the ancestral dynamic is.
So,
Questions?
in this slide, you said that time proceeds radially around this.
Yeah.
The sizes are the same?
No, the sizes actually represent how many substitutions happen, but they could happen at different rates in different species. That's why the length of this is much longer than the length of that, because really more substitutions happen here than here. I could rescale this for time also. Okay. How do we know what the ancestral mitochondrial division apparatus look like if they, all the dynamics, do different things? Just like asking humans and chimpanzees which was the ancestor. Okay. Now this is the most complicated slide. I put it up here to some extent for shock and awe, just to say that there is some really complicated stuff on that here. But also because you can actually follow this. And I'm going to spend about 5 minutes working through the logic. So the moment you know this thing down, you know all this complicated. Just look at this cartoon. This again is a radial phylogenetic. The center is T equals 0. As time proceeds, you go radially on. On that tree, things keep diverging. There are new types that are created from old types. And you can think of this as the tree of proteins, not the tree of species. The black circles are things that are alive today. They're proteins and animals that are still alive. The white circles are the things we're looking for. They're the types of proteins in some ancestral creature which was 2 billion years old, which is no longer alive. How do we find it? Question. So, suppose I were just to tell you that, just look at this type A. If I were to tell you that there's a bunch of proteins here, 1, 2, 3, well, 1, 2, 3, 4, 5. There are 5 proteins alive in present day organisms. Which one of those looks like the ancestor? How would you figure it out? Yes?
How do you sort of make this tree?
You make this tree by pairwise distances of sequences.
Of DNA sequence
So
of
we have the DNA sequence corresponding to protein, all the protein sequences.
all the proteins.
Yeah. So this is just a cartoon. Suppose you have got pairwise distance. So suppose, just look at the blue part since it's a bit clearer than the others, or the orange for that matter. Suppose you knew that 1, 2, 3, 4, 5, 6, 7 proteins were around today. How do you know which one look like the ancestor? From the tree, of course, it's obvious. The B's are the ancestors. And here, the A's are the ancestors. Of course, I label them because I know them. Yeah? But how did I label them? The way I label them is the following thing. Look at this. These proteins exist in a bunch of species. These are the species they exist. And if you notice, there is a type A protein on this side and a type A protein on that side. And so the simplest explanation is that the type A protein must have been up there. However, there is a type D protein only there, or there is a type E protein over here. Or for that matter, there are 2 F proteins, but only on this branch and not on the other side. So in this way, you can actually work backwards. All you have to do is to say, if there are 2 very similar proteins in very different species, they are the ancestors. I just walked you through this by hand waving, but you can train a computer to do this. When you do that, this is the real tree you get from real data. Okay? And among other things, you find, for example, there is a big blob corresponding to A, which contain a bunch of proteins, some of whose descendants are mitochondrial division proteins, some of whose descendants are vesicle fission proteins. But these are the ancestors. So now the question is just what does that type of protein do today? It's a protein that exists today, which is similar to the ancestor type, just like chimpanzees exist today, which are similar to the ancestor type of humans. Okay. So this is how you do it in practice. You get a bunch of sequences, and you try and reconstruct using this method. It's called parsimony to go all the way back to the ancestor. You do the vesicle scission, move from mitochondrial. You don't have to look at this at all. I'm just going to show you the summary. Remember the tree I showed you initially? Fungi, metazoans, plants, diatoms, all kinds of stuff. Remember that tree, right? All I want you to do is to look at how we label the tree. After doing all these statistics, we found the following: that today, these 4 groups actually have 2 types of dynamins in their cells. One type does the vesicle stuff, the other type does the mitochondrial stuff. That's why I've shown 2 lines, one is pink and one is blue. But these types of cells look more like this. They have one dynamin that's doing both things. Okay? So the ancient one was the jack of all trades. We have a specialized. Okay. Now that we know that the descendants look like that, you have to ask, well, suppose you didn't know the ancestry. Now how did I label the ancestor? I don't know. I don't have a time machine, so I don't know what to call the ancestor. So what is the simplest explanation for the ancestor? You can do this statistically. It turns out the answer is it's much more likely the ancestor looked like this guy than like this guy. So the ancestral dynamin was the jack of all trades dynamin. Alright. You do this by using a mathematical model. Yet another case where you need some serious, rigorous analysis in order to make these claims. I'm not just making it off the top of my head. Fine. So to cut to the chase, last slide, the last couple of slides. I advertised something about billion neural cells, right? But then I've given you a story about some protein reconstruction and so on. So you should all be like demanding satisfaction. So where's the answer? What has this got to do with reconstructing the ancestral cell? Alright. I want you to take a look at this again. I flashed it in front of you for a reason, because none of us can take in this much information quickly. But I just want you to look at mitochondria. Turns out that I lied to you. Of course, our mitochondria, we are called the crispitochondria. Okay, another crazy thing. Our mitochondria don't have FDSZ. Plant mitochondria don't have FDSZ. Paramecium mitochondria don't have FDSZ. All that is true. But there are some strange cells which do still have FDSZ in their mitochondria. Okay? And when we started working on this, it wasn't clear why some cells had that, some cells had this. Just take a look at this chart in the context of what we had discovered. Here's what we discovered. On the same tree, you may not have noticed, I put these black ticks. Those are the ticks where the FDSZ got lost. That's where it was not lost. Right? Coincidentally, the guys that still retain the bacterial division machinery in the present day also retain what we claim was the ancestral mitochondrial division machinery from the host, from the outside. All I'm saying is that these types of cells, therefore, in the present day, look just like the cells look 1.8 billion years ago. Right? I'm not making this up. I can do this by adding a lot of statistical rigor to make the claim. Which cells in the present day look like the ancestor? Not at all obvious. Once you go through all this, you say, these are the species whose mitochondrial division looks just like cells mitochondrial division apparatus would have looked if you made a time machine, went back 1.8 billion years, and brought it back to the present. So if you're really interested in the past, you're not left asking non-scientific questions. You can ask a rigorous question. So I like to make this post. Wanted for failure to evolve. Right? People think everything has to keep on evolving, right? Becoming better and better. Some things remain the same. Some parts of some things remain the same. Some parts of some things change. Nothing wrong with that. So it turns out, like I said, if you want to study what stuff was looking like 2 billion years ago, go study Streptomyces. One example are the diatoms in the ocean. Go study amoebozoans, which you must have studied in school. Go study red algae, that's seaweed. If you study all these are what mitochondria look like 2 billion years. And now I've converted a crazy question into an experimentally practical problem, which somebody can go do the experiment. And you need to be done, and you need to go for it. Okay? Fine. I just want to point out, this is not new. Of course, Darwin was known all about this. Darwin has a chapter in the Origin of Species, which you read when you finally have the time to do it. Where he speculates about how something as complicated as I could have evolved, very intermediate states. And he realizes that if you don't have a time machine, it's very hard to answer this question. So he says the following thing: in looking for the gradations by which an organ has been perfected, we ought to look exclusively to its lineal ancestors. Find out where the eye came from, look at the ancestral eye. But this is scarcely ever possible. We don't have a time machine. Fossils are usually pretty bad. We are forced in each case to look to the collateral descendants from the same original parent form, looking for guys of the ancestral eye that still survive to the present day. These anomalous forms can almost be called living fossils. They have endured to the present day. If you find anything in the present day that looks just like the ancestor, then you just have to go. That's the correct study. Sounds easy. The whole point is you never know which one it is unless you do the statistics. Okay. So this is a phylogenetic tree. Just one example of that. So it turns out when you do the statistical analysis and you make a phylogenetic tree, then you find these kinds of structures that are right in the center, which are called galls. They're called galls because they look like the galls which are diseases that plants get. I don't know how many of you have seen it. A tree has a big knot like that, right? Typically it's a disease. But that's neither here nor there. That's called a gall on a tree. This is called a gall on a phylogenetic. Whenever you see a gall, it turns out to be a statistical indication of an ancestral state. That's basically what we found. This is a mathematical version of Darwin's. So I'll stop there. Project was done by a fantastic graduate student in the lab, Ramya, and it was inspired. Krishnan was the person whose lab I went to and said about mitochondrial device. He was one of the most interesting scientists I ever knew. He passed away a few years ago. But he worked on anything and everything. He was a physicist, but he decided that biology had more than enough problems to keep him occupied for the world. And the reason we ended up doing this crazy project was really inspired by it. If you feel like getting a bit of punishment, you can actually read the papers, but you don't have to. See the talk. And more information on our website. So I'll take questions. Okay, thank you. And we had a lot of questions in the I have to take a couple more. Place where?
Time rates.
This? Yeah.
This shows that it's stabilizing. All the rates.
This shows that
All of them are stabilizing, right? All the rates.
the rates are stabilizing. No, it just turns out that every stochastic process has an equilibrium distribution. And in order to figure out the dynamics, it's often easy to figure out the distribution and then figure out the dynamics. So you'll learn about this when you do your Markov change class. This is a Markov process. This is a Markov process of change. How many of you have heard of Markov process? Yeah, so this is an example. Yeah.
So you could run this program forward and you can see what happens.
You can run it forward. You can run it forward, and you can only say what the probability of various outcomes is. Okay? But, very good question. So he wants to know, I predicted the past. That's not a big deal, right? Right? So everybody knows Yogi Berra died recently. He was a baseball hero from the United States. So he had the same. Prediction is hard, especially about the future. Yeah? So you want me to predict the future. So it turns out we did make a prediction. We predicted that it's highly unlikely for the ancestral state to survive for very long. And if you wait long enough, all the ancestors, which is the jack of all trades, will duplicate, and the true descendants will take on the specialized functions of them. And it turns out that this indeed has happened many different times. In those study also process. So we have in some sense predicted the future. In the sense that if you imagine that I restrict my attention, suppose you told me any stuff that happened up to here, I could have predicted how many branches here would have been. Not which one. Yeah. Yes, yes. So in that case, they do have much more data than you, because they have individual viruses. But they're also predicting for a much shorter time. So over such a short time, it's the time. It's like you can predict weather for 5 days, you can't predict for 7 days.
Is there some analogy to radioactive decay when some change suddenly occurs?
Sometimes it happens. In fact, we are very interested in the idea that cells, most of the time, evolve gradually, occasionally evolve in jumps. And you see two examples here. Mitochondria going into archaea is an example of a jump. So these jumps are objects which are very difficult to model using standard types of differential equations, because they represent these little delta functions in the equation, right? So it does happen, and it's a very understudied area. So it's an open question.
Can we ever have?
We can.
Can we ever have considerable amount of control over the environment to simulate something?
We can do better. We can run the evolution experiment with real cells in the lab, which is something we do. We can simulate it. You can actually see it happen. Okay, so the last burning question.
When forecast you have the dynamic integration system used
Yeah.
for viruses, how do you set up? Are they personal integrations?
So what you do is you characterize the relative rates of A to G transitions, G to C transitions, selected and non-selected regions. And then what you really want to know is, if you have a bunch of viruses that I've trained, you see a lot of closely related ones. What that says is that their ancestor for some reason is contributing disproportionately to the present. And therefore has some sort of fitness advantage. And you would therefore predict this guy much more likely to seize the next visible. So it's a purely statistical argument, but it's based on conditioning on the recent past of some model which has some other parameters. Based on probability. Everything is based on probability. If you have to learn one thing in your life, please learn about probability. Random walks. Okay, so I'll stop here and I'll let you guys go to lunch.